The ethical implications of genetic editing, particularly with technologies like CRISPR, have sparked a wide range of discussions as people delve into the potential benefits and risks of altering the genetic makeup of organisms, including humans. With the ability to edit genes, scientists can potentially eliminate genetic diseases, enhance agricultural yields, and even create organisms with desirable traits. However, these advancements raise significant ethical questions about the extent to which we should manipulate life itself. One of the main concerns revolves around the concept of playing God, where critics argue that humans might overstep natural boundaries, leading to unforeseen consequences on ecosystems and society as a whole. Furthermore, the issue of consent becomes particularly murky when considering germline editing, which affects not just the individual being edited, but also future generations, raising questions about the rights of those who cannot consent to such modifications. There is also the fear of creating a genetic divide, where those who can afford genetic enhancements might gain unfair advantages over those who cannot, leading to increased social inequality. Additionally, the possibility of unintended consequences, such as off-target effects that could introduce new diseases or traits, adds another layer of complexity to the debate, emphasizing the need for thorough research and regulation before widespread application. Moreover, the cultural and religious perspectives on genetic editing vary widely, with some viewing it as a potential pathway to improving human health, and others seeing it as a violation of natural laws or divine principles. As we explore the frontiers of genetic science, it becomes increasingly important to engage in open conversations about these ethical implications, ensuring that scientific progress aligns with our moral values and social responsibilities. The balance between innovation and caution is a delicate one, as we weigh the potential benefits of genetic editing against the risks and ethical dilemmas it presents, marking a pivotal moment in our understanding of genetics and its place in our lives. CRISPR technology, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, has taken the scientific world by storm, capturing the attention of researchers, biologists, and even the general public due to its revolutionary ability to edit genes with unprecedented precision and efficiency. The discovery of CRISPR's role in the immune systems of bacteria, where it functions as a defense mechanism against viruses, has paved the way for its adaptation in genetic engineering. This technology allows scientists to target specific sequences of DNA, making it possible to remove, add, or alter genetic material in a wide range of organisms, from plants to animals and even humans. One of the most exciting applications of CRISPR is in agriculture, where it is being used to develop crops that are resistant to pests, diseases, and even adverse environmental conditions, which could lead to increased food security as the global population continues to grow. Additionally, CRISPR is making waves in the medical field, where it holds the potential to treat genetic disorders such as cystic fibrosis, sickle cell anemia, and muscular dystrophy. In these cases, researchers are exploring the possibility of correcting mutations at the genetic level, offering hope for effective treatments that could change the lives of countless patients. Beyond treating genetic disorders, CRISPR is also being investigated for its potential in combating infectious diseases, including HIV and even some forms of cancer, as scientists work to harness its capability to disrupt the genes that drive these illnesses. However, while the possibilities are thrilling, the ethical implications of CRISPR technology are equally significant, raising questions about the potential for designer babies, gene editing in embryos, and the long-term effects of altering the human genome. As researchers continue to explore these possibilities, the regulatory landscape surrounding CRISPR technology is also evolving, with governments and organizations striving to establish guidelines that balance innovation with safety and ethical considerations. The ongoing advancements in CRISPR technology, coupled with its diverse applications, highlight a transformative era in genetic research, where the boundaries of science are continually being pushed, leading to groundbreaking discoveries and innovations that could reshape our understanding of biology and medicine. Designer babies, A concept that once seemed purely science fiction is now becoming a reality thanks to advancements in genetic engineering and reproductive technologies. The idea of designing a baby, where parents can choose specific traits such as eye color, intelligence, or even resistance to certain diseases, raises numerous ethical, social, and medical questions. On one hand, the pros of designer babies are quite compelling. For instance, the ability to eliminate hereditary diseases can potentially lead to healthier lives for future generations. Imagine a world where conditions like cystic fibrosis, sickle cell anemia, or even certain types of cancer could be edited out of the genetic code, reducing suffering and healthcare costs significantly. Moreover, the prospect of enhancing human capabilities, whether it be cognitive functions or physical attributes, could mean a population that is not only healthier, but also more capable, pushing the boundaries of human achievement. However, these benefits come with notable cons that cannot be overlooked. The moral implications of playing God stir deep philosophical debates about what it means to be human. Critics argue that designer babies could widen the gap between socioeconomic classes, as only those who can afford such technologies may benefit from them, leading to a society where genetic enhancements become a privilege for the wealthy. This could foster a new kind of inequality, where a genetic elite emerges, exacerbating existing social divides. Additionally, the long-term effects of genetic modifications are still largely unknown. While we can make educated guesses about certain traits, the interplay of genes is incredibly complex, and unintended consequences could arise, potentially leading to unforeseen health issues or genetic anomalies. Moreover, there is also the concern of consent. A designer baby, whose traits have been predetermined by its parents, may one day question its identity, feeling the weight of expectations placed upon it. The psychological effects of being a custom-made individual could have profound implications on personal development and self-worth. As research progresses and technologies like CRISPR continue to evolve, the discussion surrounding designer babies becomes increasingly urgent. The ethical frameworks we establish now will shape the future of human reproduction, and society must grapple with these intricate dilemmas. The balance between scientific potential and ethical responsibility is delicate, demanding careful consideration and open dialogue among scientists, ethicists, and the public alike. Genetic privacy and ownership issues are becoming increasingly significant in our modern world, as advancements in genetic testing and biotechnology raise complex ethical questions. With the rise of direct-to-consumer genetic testing, many individuals are eager to explore their ancestry, health risks, and even traits, yet they might not fully understand the implications of sharing their genetic data. When someone submits their DNA to a testing company, they often agree to terms of service that can include giving the company the right to use their genetic information for research, development, or even commercial purposes. This raises important questions about who truly owns that genetic information, and whether individuals can control how it is used once it leaves their hands. Furthermore, privacy concerns are paramount, as genetic data can reveal sensitive information not just about an individual, but also about their relatives. For instance, if a person discovers they have a genetic predisposition for a certain disease, that information could potentially impact their family members who may share similar genetic traits. This leads to a dilemma regarding informed consent, as not all relatives may wish to have their genetic information exposed or utilized for research without their explicit consent. Moreover, the potential for genetic discrimination looms large in the background of these discussions. Employers and insurance companies could theoretically use genetic data to make decisions regarding hiring, promotions, or coverage, which raises the risk of unfair treatment based on genetic predispositions rather than actual health status. While laws like the Genetic Information Non-Discrimination Act in the United States aim to protect individuals from discrimination based on genetic information, the landscape is still fraught with uncertainty. As technology continues to advance faster than legislation can adapt, the implications of genetic ownership also extend into the realm of intellectual property. As scientists explore gene editing technologies, such as CRISPR, the question of whether specific genetic sequences can be patented or owned becomes crucial. If a company can patent a gene associated with a particular trait or condition, this could create monopolies over certain genetic technologies, which raises ethical concerns about access to these innovations and the potential for exploitation. In addition, the global nature of genetic research complicates ownership issues further. Genetic samples collected in one country might be analyzed in another, leading to questions about jurisdiction and the rights of individuals whose genetic data crosses borders. Different countries have varying regulations concerning genetic privacy and ownership, which can create a patchwork of protections that may not adequately safeguard individuals' rights on an international scale. As we continue to navigate the waters of genetic privacy and ownership, public, regulatory bodies play a crucial role in the field of genetic engineering, overseeing the safety, efficacy, and ethical implications of genetic modifications. These organizations, which vary by country, are tasked with ensuring that genetic engineering practices do not pose risks to human health, animal welfare, or the environment. In the United States, for example, the Food and Drug Administration, or FDA, evaluates genetically engineered products, including food and drugs, to ensure they are safe for consumption and effective for their intended use. Similarly, the Environmental Protection Agency, or EPA, regulates genetically modified organisms, particularly those that might affect the environment, such as genetically modified crops that could potentially impact local ecosystems. In Europe, the European Food Safety Authority, or EFSA, plays a significant role in assessing the safety of genetically modified foods, providing scientific advice to policymakers, and ensuring that the public is protected from any potential hazards associated with genetic engineering. The regulations set by these bodies often require extensive testing and evaluation before new genetically modified products can be approved for public use. This thorough review process includes analyzing potential health risks, environmental impacts, and long-term effects on biodiversity. Moreover, regulatory bodies also establish guidelines for labeling genetically modified foods, which helps consumers make informed choices about what they are purchasing and consuming. In some countries, regulations are more stringent, requiring clear labeling of any products that contain genetically modified ingredients, while in others, the rules may be less strict, leading to ongoing debates about transparency and consumer rights. Ethics also play a significant role in genetic engineering regulations. Regulatory bodies often grapple with complex ethical questions, such as the implications of gene editing in humans, particularly with technologies like CRISPR. In some regions, there are strict guidelines that prevent the use of genetic engineering for human enhancements, while in others, discussions are ongoing about the boundaries of acceptable research and application. This ethical oversight is crucial in ensuring that advancements in genetic engineering do not lead to unintended consequences, such as exacerbating social inequalities or creating new forms of discrimination. Additionally, international cooperation among regulatory bodies is becoming increasingly important, as genetic engineering technologies are not confined by national borders. Organizations such as the World Health Organization, or WHO, and the Food and Agriculture Organization, or FAO, work to develop global standards and guidelines for genetic engineering practices, addressing health and safety concerns on an international scale. This collaboration helps to ensure that countries can share knowledge, resources, and best practices, while also addressing the potential risks associated with genetic engineering technologies. Gene therapy is a fascinating and rapidly evolving field in medicine, aiming to treat or even cure genetic disorders by directly manipulating the genes responsible for the disease. The fundamental idea behind gene therapy is to replace, inactivate, or introduce genes into a patient's cells to correct genetic abnormalities. For instance, one of the most groundbreaking applications of gene therapy has been in treating inherited conditions like cystic fibrosis and hemophilia, where defective genes lead to severe health issues. Researchers have developed various methods to deliver therapeutic genes into cells, including viral vectors, which are modified viruses that can carry new genetic material. These vectors are designed to be safe and effective, allowing the healthy gene to enter the patient's cells and produce the necessary proteins that the body lacks due to the genetic disorder. Additionally, there have been significant advancements in using CRISPR technology, a revolutionary tool that allows scientists to edit genes with precision. This technique has opened up new possibilities, such as the potential to eliminate genetic diseases by directly correcting mutations at their source. For example, in clinical trials, researchers have successfully used CRISPR to treat conditions like sickle cell disease, where specific genetic alterations can restore normal blood cell function. The precision of CRISPR not only enhances the effectiveness of the treatment, but also minimizes the risk of unintended effects, making it a promising option for future therapies. Moreover, gene therapy is not limited to inherited disorders. It also shows great promise in treating cancers, where genetic mutations drive the growth of tumors. By using gene therapy, doctors can introduce genes that help the immune system recognize and attack cancer cells more effectively. For instance, CAR T-cell therapy, a type of gene therapy, involves modifying a patient's T cells to better target and destroy cancer cells. This approach has already transformed the treatment landscape for certain types of blood cancers, providing hope to patients who previously had few options. Despite its potential, gene therapy is not without challenges and ethical considerations. The long-term effects of gene therapy are still being studied, and there are concerns about the possibility of unintended consequences, such as off-target effects, where the gene editing might inadvertently affect other genes. Additionally, the cost of gene therapy treatments can be prohibitively high, raising questions about accessibility and equity in healthcare. Ongoing research aims to address these challenges, ensuring that gene therapy can be a viable option for a wider range of patients. As the field of gene therapy continues to advance, we are likely to see more innovative treatments emerging, potentially leading to cures for previously. The environmental impact of genetically modified organisms, often referred to as GMOs, is a topic that generates a lot of discussion and debate. GMOs are organisms whose genetic material has been altered using genetic engineering techniques, and they are commonly used in agriculture to enhance crop yield, resistance to pests, and tolerance to herbicides. One of the significant environmental benefits of GMOs is their potential to reduce the need for chemical pesticides. For instance, some genetically modified crops have been engineered to express proteins from the bacterium Bacillus thuringiensis, which can naturally kill certain pests, thus lowering the reliance on synthetic pesticides that can harm non-target organisms and pollinators, like bees. This reduction in chemical use can lead to healthier ecosystems, as fewer chemicals mean less runoff into waterways, which can cause issues like algal blooms that deplete oxygen and harm aquatic life. However, the introduction of GMOs into the environment is not without concerns. One major worry is the potential for crossbreeding with wild relatives, which can lead to the creation of superweeds that are resistant to herbicides. This phenomenon could result in increased herbicide usage, which may counteract some of the benefits that GMOs provide. Additionally, there are concerns about the impact on biodiversity. The widespread cultivation of a few genetically modified crop varieties may lead to a reduction in genetic diversity in agricultural systems, making them more vulnerable to diseases and pests. This reliance on monoculture, where a single crop is grown extensively, can also affect soil health and ecosystem resilience, leading to greater susceptibility to climate change impacts. Moreover, the environmental impact of GMOs can vary based on farming practices and local ecosystems. For example, in regions where GMOs are used, the introduction of beneficial insects and natural pest control methods can be diminished, as the ecosystem becomes more dependent on genetically engineered traits rather than on natural ecological balances. Furthermore, there is ongoing research into the long-term effects of GMOs on soil health, with some studies suggesting that certain genetically modified crops could alter soil microbial communities, which play a crucial role in nutrient cycling and plant health. On the flip side, proponents argue that GMOs can contribute to sustainable agriculture. By enabling farmers to grow crops that require fewer resources, such as water and fertilizers, GMOs can help mitigate the effects of climate change and food scarcity. For instance, drought-resistant crops can thrive in challenging conditions, allowing farmers to produce food even during dry spells, which is increasingly important in the context of the concept of playing God in genetics brings to mind a fascinating and complex debate about the power humans have over life itself. As genetic technology advances at an unprecedented pace, scientists are now able to alter the building blocks of life, leading to significant ethical questions and philosophical discussions. For instance, the development of CRISPR-Cas9, a revolutionary gene editing tool, allows researchers to make precise changes in DNA, potentially curing genetic diseases, enhancing crop resilience, and even combating climate change by altering the genetics of certain species. However, this power also raises concerns about unintended consequences, such as the creation of genetically modified organisms that could disrupt ecosystems, or the ethical implications of editing human embryos, which some see as crossing a moral line. In the realm of human genetics, discussions about playing God often center on the idea of designer babies, where parents might choose specific traits for their children, such as intelligence, physical abilities, or even appearance. This notion brings forth a myriad of ethical dilemmas, including issues of inequality, as those with access to genetic modifications might create a divide between the genetically enhanced and those who are not. The potential for eugenics, or selective breeding, echoes darker periods of history, stirring fears about discrimination and the loss of genetic diversity. Moreover, the concept of playing God is not limited to humans. It extends to the animal kingdom as well. Scientists have used genetic editing to create disease-resistant livestock, which could greatly benefit food security, but this manipulation also prompts questions about animal welfare and the natural order of species. The idea of humans intervening in nature raises significant concerns about arrogance and the consequences of assuming a godlike role in shaping life. Additionally, the use of genetic technology in agriculture has sparked debates over food safety, environmental impact, and the rights of farmers. Genetically modified crops, designed to resist pests or tolerate harsh conditions, can lead to higher yields and reduced pesticide use, but they can also create dependence on specific seed companies, which can monopolize the market and affect food sovereignty. The complexities of these discussions highlight the need for responsible stewardship of genetic technologies, balancing innovation with ethical considerations. As we venture further into the realm of genetics, the question remains, where do we draw the line? Are we prepared to accept the potential consequences of our actions? And can we ensure that our advancements in genetic engineering benefit all of humanity rather than just a privileged few? The interplay of science, ethics, and society continues to. Genetic manipulation, also known as genetic engineering, has made remarkable strides in recent decades, allowing scientists to alter the DNA of organisms, leading to advancements in medicine, agriculture, and even environmental conservation. However, despite its potential, there are significant limits to what can be achieved through genetic manipulation. One of the primary concerns is the complexity of genetic systems. The interactions between genes, their expressions, and the environment create a web of influences that are not fully understood, making it challenging to predict the outcomes of genetic modifications accurately. For instance, while scientists can introduce a gene that confers resistance to a disease in a crop, the broader ecological effects of that modification can be unpredictable, potentially affecting surrounding plants, insects, and soil health in ways that are not immediately apparent. Another limitation is the ethical landscape surrounding genetic manipulation. The debate about genetically modified organisms, or GMOs, is ongoing, with concerns regarding food safety, environmental impact, and the rights of consumers to know what is in their food. Many people worry about the long-term effects of consuming genetically modified foods, even though numerous studies have shown that they are safe for consumption. Additionally, there are ethical questions surrounding the manipulation of human genes, especially when it comes to editing genes that could be passed down to future generations. The idea of designer babies, where parents could choose traits for their children, raises moral dilemmas that society is still grappling with. Regulatory frameworks also impose limits on genetic manipulation. Different countries have varying policies regarding the approval and use of genetically modified organisms. In some regions, the process is heavily regulated, requiring extensive testing and evaluation before any genetically modified product can reach the market, while in others, the regulations are more lenient. This inconsistency can slow down innovation, as researchers may find it difficult to navigate the different legal landscapes across borders. Moreover, technical limitations exist within the field of genetic engineering itself. Techniques like CRISPR-Cas9 have revolutionized the way genes can be edited, allowing for precise changes. However, off-target effects, where unintended parts of the genome are altered, can occur, leading to potentially harmful consequences. The technology is still evolving, and while it holds enormous promise, it is not infallible. Researchers continue to work on improving these techniques to minimize risks and enhance precision. Finally, there is the issue of public perception and acceptance. Even with a solid scientific foundation, if the public is not on board with genetic manipulation, it can hinder progress. Education, genetic discrimination, a term that refers to unfair treatment based on an individual's genetic information, has become an increasingly important topic, especially in the realms of employment and insurance. As advancements in genetic testing and research continue to grow, so does the potential for misuse of genetic data. For instance, some employers might be tempted to use genetic information to predict an employee's future health risks, potentially leading to discriminatory practices that could affect hiring decisions, promotions, or job security. Imagine applying for a job, only to find out that an employer has accessed your genetic data and decided against hiring you, fearing that you may develop a costly health condition in the future. This kind of scenario raises significant ethical concerns, and it has prompted many to call for stricter regulations. In the United States, the Genetic Information Non-Discrimination Act, or GINA, was enacted in 2008 to address these concerns. GINA prohibits discrimination in health insurance and employment based on genetic information, protecting individuals from being treated differently due to their genetic predispositions. However, gaps still exist, as GINA does not cover life insurance, long-term care insurance, or disability insurance, which means individuals may still face discrimination in those areas. This leaves many feeling vulnerable, as they may be unable to access essential services simply because of their genetic profile. Across the globe, various countries have taken different approaches to tackle genetic discrimination. For example, the European Union has implemented regulations that protect individuals from discrimination based on genetic data. But the enforcement and effectiveness of these regulations can vary widely among member states. As more people undergo genetic testing, the discussions surrounding privacy, consent, and discrimination become increasingly vital. The potential for insurers to deny coverage based on genetic predispositions is a pressing issue, as it could lead to people avoiding necessary genetic tests out of fear of discrimination. Moreover, the social implications of genetic discrimination can be profound. As society becomes more aware of genetic risks, individuals may feel pressure to conform to certain health standards, leading to a culture of stigma surrounding those with certain genetic markers. This stigma can affect mental health, as individuals may experience anxiety or depression due to the fear of discrimination. It is crucial to foster an environment of understanding and acceptance, where genetic information is used responsibly and ethically. In the context of employment, the handling of genetic information must be approached with caution, as it may influence workplace dynamics. companies need to cultivate inclusive cultures that do not penalize employees for their genetic makeup, ensuring that such information, public perception of genetic engineering varies widely, influenced by cultural beliefs, media representation, and individual understanding of science. Many people, when they hear the term genetic engineering, immediately think of controversial topics like genetically modified organisms or GMOs in food production. Some view genetic engineering as a way to enhance food security and combat hunger, while others fear potential health risks and environmental consequences. In fact, according to various surveys, a significant portion of the population expresses concern about the long-term effects of consuming genetically modified foods, fearing that it may lead to unforeseen health issues or ecological damage. On the other hand, there's a growing segment of the public that embraces genetic engineering, particularly in the context of medical advancements. Techniques like CRISPR, which allow for precise editing of DNA, have sparked excitement about their potential to cure genetic disorders, treat diseases, and even enhance human capabilities. This enthusiasm is often bolstered by success stories in gene therapy, where patients have experienced remarkable recoveries from previously untreatable conditions. However, the idea of designer babies, where parents could select traits for their children, raises ethical questions that many people find unsettling. This concept leads to debates about the moral implications of playing God and the potential for societal divides based on genetic enhancements. Additionally, public perception is heavily shaped by media portrayals, which can often skew towards sensationalism. Documentaries, movies, and news stories sometimes highlight worst-case scenarios, leading to increased fear and skepticism. Conversely, educational programs and articles that explain the science behind genetic engineering in an accessible manner can help demystify the topic, leading to a more informed public. Research shows that when people understand the science and its benefits, they are more likely to support genetic engineering initiatives, particularly in healthcare. Another layer to this perception is the influence of trust in regulatory bodies and scientific communities. When people feel confident that there are strict regulations and oversight in place, they are generally more supportive of genetic engineering. Trust in scientists and institutions can significantly sway opinions, as transparency and clear communication about the methods and intentions of genetic engineering are crucial. Surveys indicate that people are more likely to support genetic engineering when they believe that scientists prioritize safety and ethical considerations. Social media also plays a significant role in shaping public opinion, as it allows for the rapid spread of information, both accurate and inaccurate. Misinformation can lead to public fear, while viral success stories can foster acceptance and enthusiasm for genetic engineering. This dual-edged sword means that in recent years, gene editing has become a hot topic, not just in science and medicine, but also in religion and ethics. The ability to edit genes, using techniques like CRISPR, allows scientists to make precise changes to DNA, which can lead to breakthroughs in treating diseases, improving crops, and even potentially enhancing human abilities. However, this powerful technology raises significant questions that intersect with religious beliefs, particularly regarding the sanctity of life and what it means to be human. Many religious groups express concern that gene editing could be seen as humans playing God, altering the very fabric of life itself, which is a concept that can be unsettling for many. For instance, some Christian denominations argue that life is sacred and that humans should not interfere with God's creation, fearing that gene editing could lead to moral and ethical dilemmas, such as designer babies, where parents might select traits like intelligence or physical appearance for their children. On the other hand, some religious leaders and organizations advocate for a more nuanced approach, arguing that gene editing could be used for good, such as eradicating genetic diseases that cause suffering. They suggest that if gene editing can alleviate pain and save lives, it could be seen as a way to fulfill a moral obligation to help fellow human beings. This perspective emphasizes the importance of intention behind the technology, proposing that gene editing should be approached thoughtfully with an ethical framework that respects human dignity and life. Judaism also offers a unique perspective on the matter, where the preservation of life is a core value. Many Jewish scholars argue that if gene editing can prevent suffering or save lives, it aligns with the Jewish principle of pikuach nefesh, which prioritizes saving lives over almost all other religious rules. Nevertheless, there is a cautious approach to the idea of altering human DNA, as it raises questions about what it means to be human and the potential for unforeseen consequences. Islamic perspectives on gene editing vary, with some scholars expressing acceptance, arguing that it can be a form of stewardship over God's creation, while others warn against altering what has been divinely ordained. The debate often centers on the balance between the potential benefits of gene editing, such as curing hereditary diseases, and the ethical implications of modifying the human genome, which could lead to changes that may not align with Islamic teachings. Furthermore, the intersection of religion and gene editing also brings to light issues of social justice. There is a fear that access to gene editing technology could be limited to wealthy individuals or nations, creating a genetic patents and intellectual property rights represent a fascinating intersection of science, ethics, and law, which has become increasingly relevant in our rapidly advancing world of biotechnology. At the heart of this issue lies the question of whether or not the products of nature, such as genes and genetic sequences, can be owned and patented by individuals or organizations. In the late 20th century, the U.S. Patent and Trademark Office began allowing the patenting of isolated genes, which sparked a heated debate over the ownership of genetic material. Proponents argue that patenting genes incentivizes research and innovation, allowing companies to recoup the costs of their investments in developing new treatments and technologies. By granting exclusive rights to inventors, patents can encourage the discovery of life-saving drugs and therapies, ultimately benefiting society as a whole. However, critics raise serious concerns about the implications of genetic patents, particularly regarding accessibility and equity in healthcare. When companies hold patents on essential genes, they can impose high prices on tests and treatments, making them unaffordable for many people. This situation can create disparities in health outcomes, particularly for marginalized communities who may not have the same access to innovative therapies. Moreover, questions arise about the ethical implications of owning parts of the human genome, as some argue that it should not be possible to claim ownership over something that is inherently part of human biology. The case of Myriad Genetics, which patented the BRCA1 and BRCA2 genes linked to breast and ovarian cancer, brought these issues to the forefront when the Supreme Court ruled in 2013 that naturally occurring genes could not be patented, although synthetically created variations could be. This landmark decision highlighted the need for a careful balance between protecting intellectual property and ensuring public access to vital medical information. In addition, the rise of CRISPR technology, which allows for precise editing of genetic material, has further complicated the landscape of genetic patents. As researchers and companies explore the potential of gene editing, the question of who holds the rights to these new innovations becomes even more critical. There are ongoing debates about whether the techniques used in gene editing can be patented, and if so, how those patents might affect future research and development. As the field of genetics continues to evolve, so do the legal frameworks surrounding intellectual property rights, leading to a complex web of regulations that can vary significantly between countries. Many nations have adopted different approaches to genetic patents, with some being more permissive than others, resulting in a patchwork of laws that researchers. The evolution of genetic engineering technology has been nothing short of remarkable. Starting in the early 1970s, when scientists first discovered how to cut and splice DNA, paving the way for what we now call recombinant DNA technology. This breakthrough allowed researchers to combine DNA from different organisms, creating genetically modified organisms, or GMOs, for the first time. One of the earliest successes in this field came in 1973, when Paul Berg, a biochemist, constructed the first recombinant DNA molecules, leading to a series of experiments that ultimately produced the first genetically engineered bacteria, which could produce insulin, a game-changer for diabetes treatment. This innovation not only demonstrated the potential of genetic engineering, but also sparked interest and investment in biotechnology, which has continued to grow over the decades. As the technology advanced, the 1980s and 1990s saw the rise of genetically modified crops, which promised to increase agricultural yields and reduce the need for pesticides. Crops like Bt corn and Roundup Ready soybeans were engineered to be resistant to pests and herbicides, respectively, significantly impacting farming practices and food production. Yet, this also raised ethical and environmental concerns, leading to debates about food safety and biodiversity, as well as the implications of patenting life forms. Fast forward to the early 21st century, the introduction of CRISPR-Cas9 technology in 2012 marked a revolutionary leap in genetic engineering, allowing for precise editing of genes with unprecedented ease and accuracy. This tool, derived from a natural defense mechanism in bacteria, enables scientists to cut DNA at specific locations and make targeted modifications, opening up possibilities not just for agriculture, but also for medicine, as researchers began exploring its potential for treating genetic disorders, cancer, and even HIV. Moreover, gene therapy has evolved significantly, with early trials facing numerous challenges, yet recent advancements have led to successful treatments for conditions like spinal muscular atrophy and certain types of inherited blindness. As technology continues to improve, the future of genetic engineering holds promise for curing diseases that were once thought to be untreatable. Additionally, ethical discussions surrounding genetic engineering have become increasingly complex, with questions about designer babies, gene editing in embryos, and the potential for unforeseen consequences. The concept of gene drives, which could spread genetic changes through wild populations, raises concerns about ecological impacts and biodiversity, prompting calls for responsible oversight and regulation. In the realm of synthetic biology, scientists are now designing and constructing new biological parts, devices. The potential for bioweapons using genetic engineering is a topic that raises many ethical, scientific, and security concerns. Genetic engineering, which involves modifying the DNA of organisms, has made incredible advancements in recent years, allowing scientists to create new strains of bacteria, viruses, and even plants. While these advancements hold great promise for medicine and agriculture, they also pose risks when applied in harmful ways. For instance, the ability to edit genes using techniques like CRISPR-Cas9 has made it easier to potentially create pathogens that are more virulent or resistant to treatments, which could be weaponized. Some experts worry that rogue states or even terrorist groups could exploit this technology to create biological agents that could be used in warfare or bioterrorism, leading to widespread disease and chaos. Historical instances, such as the use of anthrax and smallpox in warfare, highlight how devastating bioweapons can be, and with genetic engineering, the ability to tailor these agents for specific targets might become a reality. Moreover, the dual-use nature of this technology complicates the situation further. While genetic engineering can lead to beneficial outcomes, it can also be misused for malicious purposes. Additionally, the global community faces challenges in regulating and monitoring genetic research to prevent such misuse, as the knowledge and tools are becoming more accessible to individuals and groups with nefarious intentions. The ethical implications are profound, as scientists must navigate the thin line between innovation and potential harm, raising questions about how much oversight is necessary to ensure that advancements in genetic engineering do not lead to catastrophic consequences. As this field continues to evolve, it is crucial for policymakers, scientists, and society to engage in conversations about the responsible use of genetic technologies, ensuring that the benefits are harnessed while minimizing the risks associated with their potential use as bioweapons. Cloning, a a fascinating yet complex topic, raises numerous ethical considerations that spark debate among scientists, ethicists, and the general public alike. At its core, cloning involves creating a genetically identical copy of an organism, which can be seen in various forms, from therapeutic cloning aimed at medical advancements, to reproductive cloning, which has gained notoriety through discussions surrounding animals like Dolly the sheep, the first mammal to be cloned from an adult somatic cell. One major ethical concern revolves around the idea of identity and individuality. If an organism is cloned, questions arise about the rights and status of that clone, especially when considering human cloning. Would the clone have the same rights as a naturally conceived person? Would it be viewed as a person or merely a product of technology? This uncertainty leads to intense discussions about the moral implications of creating life in such a manner. Another critical ethical consideration pertains to the potential for suffering. Cloning processes often involve a high rate of failure, leading to numerous embryos that do not survive the cloning process or suffer from genetic abnormalities. This raises concerns about the welfare of animals in research, as many cloned animals experience health problems and shorter lifespans. The debate intensifies when considering the implications for human cloning, where the risks to the health of clones could be significant, raising questions of whether it is ethical to pursue such technology if it may lead to suffering. Furthermore, the concept of playing God often emerges in discussions about cloning. Many individuals hold strong beliefs that manipulating the process of life and reproduction crosses moral boundaries. This perspective emphasizes the sanctity of life and the natural order, suggesting that humans should not interfere with the biological processes that have evolved over millions of years. Ethical frameworks, such as utilitarianism, which focuses on the greatest good for the greatest number, come into play as people weigh the potential benefits of cloning, such as advances in medicine, organ regeneration, and the preservation of endangered species against the moral quandaries it presents. Issues of consent also arise, particularly in the context of cloning humans. Since a clone cannot consent to its own creation, ethical questions surface regarding the rights of the individual being created. Should parents have the right to bring a clone into existence for their own desires or needs, such as replacing a lost child or providing organs for transplantation? These scenarios provoke deep reflections on autonomy and the rights of future generations. Additionally, the implications of cloning on genetic diversity cannot be overlooked. Cloning could potentially lead to a reduction in genetic engineering. A fascinating field of science has made significant strides in both agriculture and medicine, each area harnessing the power of this technology to address specific challenges. In agriculture, genetic engineering is often used to create crops that are resistant to pests, diseases, and harsh environmental conditions. For instance, scientists have developed genetically modified organisms, or GMOs, which can withstand drought, reducing the need for water and helping farmers maintain yields even in challenging climates. This is particularly important as climate change continues to impact global food production. Moreover, genetic engineering can enhance nutritional content, as seen in the development of golden rice, which is enriched with vitamin A to combat malnutrition in regions where rice is a staple food. This approach can lead to improved health outcomes in populations that rely heavily on specific crops for their diet. On the other hand, in the field of medicine, genetic engineering plays a crucial role in developing therapies and treatments for various diseases. One of the most groundbreaking advancements is gene therapy, which involves altering the genes inside a person's cells to treat or prevent disease. For example, scientists are working on using gene editing techniques like CRISPR to target and correct genetic mutations that cause conditions such as cystic fibrosis or sickle cell anemia. This revolutionary approach holds the potential to cure genetic disorders rather than just managing symptoms, offering hope to many patients and their families. Additionally, genetic engineering is instrumental in producing biopharmaceuticals, including insulin and monoclonal antibodies, which are used to treat a variety of illnesses, such as diabetes and certain cancers. While both sectors utilize genetic engineering, the ethical considerations and public perceptions can differ greatly. In agriculture, concerns often revolve around the safety of GMOs for human consumption and their environmental impact, such as the potential for reduced biodiversity and the emergence of pesticide-resistant pests. In contrast, medical applications often focus on the ethical implications of gene editing, especially regarding germline modifications that could be passed down to future generations. This has sparked heated debates about the extent to which we should intervene in human genetics, raising questions about safety, consent, and the long-term consequences of such technologies. Furthermore, the regulatory frameworks for these applications also vary widely, with agricultural GMOs facing rigorous testing and approval processes, while medical genetic engineering, particularly gene therapies, must navigate a complex landscape of clinical trials and ethical oversight. Public acceptance plays a significant role in the progress of both fields, as perceptions of safety and ethicality can influence policy decisions and research funding. Germline editing and somatic editing are two different approaches to genetic modification, each with unique implications and applications. Germline editing involves making changes to the genes in germ cells, which are the reproductive cells like sperm and eggs, meaning that any modifications made will be passed down to future generations. This type of editing has the potential to eliminate genetic diseases from a family line, but it raises significant ethical questions, particularly regarding the long-term effects on the human gene pool and the possibility of unintended consequences that could arise from altering the very building blocks of life. In contrast, somatic editing targets non-reproductive cells, meaning that any changes made affect only the individual and cannot be inherited by their offspring. This approach is often seen as less controversial, as it offers the possibility of treating diseases such as cancer or genetic disorders without the ethical dilemmas posed by altering the germline. However, somatic editing also comes with its own set of challenges, including ensuring that the edits are precise and do not inadvertently affect other genes, a process known as off-target effects. Both methods utilize advanced technologies like CRISPR-Cas9, which allows for precise cuts in DNA, but the implications of their use are vastly different. While germline editing could theoretically lead to designer babies, where genetic traits could be selected, somatic editing is primarily focused on therapeutic applications, bringing hope to patients suffering from debilitating conditions. The ongoing debate surrounding these two forms of editing touches on scientific, ethical, and societal dimensions, as researchers, ethicists, and the public grapple with the potential to reshape human health and evolution itself. As our understanding of human genetics continues to evolve, so too does the conversation about the responsible use of such powerful technologies, and finding a balance between innovation and ethical responsibility remains a challenging frontier in the world of genetic science. The future of human evolution, particularly with the advancements in genetic engineering, opens up a fascinating realm of possibilities as scientists and researchers explore how we can directly influence our genetic makeup. Genetic engineering, which involves altering the DNA of an organism, has already shown potential in various fields, from agriculture to medicine, and now it could redefine what it means to be human. With technologies like CRISPR-Cas9, which allows precise editing of genes, the ability to potentially eradicate genetic disorders, enhance physical abilities, or even alter traits such as intelligence and longevity, is becoming more attainable. However, this raises significant ethical questions, as the idea of designer babies becomes more plausible. What does it mean to choose specific traits for our children? The implications of such choices could lead to a society where inequality is exacerbated, as only those who can afford these modifications may benefit from enhanced capabilities. In addition to health and physical traits, genetic engineering could impact our cognitive abilities, leading us to ponder the future of intelligence. Imagine a world where learning capabilities are optimized through genetic modifications, potentially leading to significant advancements in education and innovation. However, the potential for misuse, such as creating a divide between genetically modified individuals and those who remain unmodified, is a concern that cannot be overlooked. As we venture into this uncharted territory, the balance between progress and ethical considerations becomes increasingly crucial. Moreover, the environmental challenges we face, such as climate change and resource scarcity, may also drive the need for genetic engineering in humans. As the planet changes, could we adapt ourselves genetically to thrive in new conditions? Some researchers speculate that future humans might possess genetic traits that allow them to withstand harsher climates or even to utilize resources more efficiently. This adaptive evolution could become a vital part of our survival strategy, but at what cost to our natural evolution and the diversity of the human experience? Another exciting aspect of genetic engineering is the potential it holds for combating aging. Scientists are studying the genes associated with longevity and cellular regeneration, aiming to unlock the secrets to a longer, healthier life. If we could manipulate our genetic code to slow down aging processes, the implications for society would be profound. Imagine a future where aging is not seen as an inevitable decline, but rather a condition that can be managed or even reversed. While this may sound like science fiction, research in this area is advancing rapidly, raising questions about the quality of life, the nature of aging, and how society would adapt to longer lifespan. The psychological impacts of genetic modification on individuals can be quite profound and complex, affecting various aspects of identity, self-perception, and societal interaction. When we consider genetic modification, particularly in the context of genetic engineering and biotechnology, it's essential to recognize that this technology not only alters physical traits, but can also influence mental and emotional well-being. Individuals who are genetically modified, or those who are aware of genetic interventions in their family or community, often experience a range of feelings, from empowerment to anxiety. On one hand, some may feel a sense of pride and optimism, believing that genetic modifications could enhance their abilities, improve health, or increase longevity. This sense of improved potential can lead to heightened self-esteem, as people may feel they possess advantages over their peers. On the other hand, the very idea of genetic modification can evoke fears and anxieties. Concerns about what it means to be natural versus modified may lead to identity crises, as individuals grapple with the implications of their altered genetics. Questions about authenticity and the essence of being human can arise, leading to feelings of alienation or disconnection from one's own sense of self. Furthermore, societal perceptions and stigmas associated with genetic modification can add another layer of complexity. Individuals may face discrimination or judgment, either from their peers or broader society, which can lead to social anxiety and isolation. The fear of being labeled as different or other can be a heavy burden to bear, impacting mental health significantly. Additionally, there are ethical implications that weigh on the minds of those aware of genetic modification, such as concerns about consent, particularly in cases where genetic changes are made before a person is born. This can lead to feelings of helplessness or lack of control over one's own life and future. The psychological burden can be compounded by the knowledge that genetic modifications may carry unknown risks or long-term consequences, leading to worry about health issues or unforeseen side effects. In the context of families, the dynamics can shift dramatically. Parents who choose to genetically modify their children might feel pressure to ensure those modifications result in success, creating unrealistic expectations. This pressure can trickle down, leading to stress and anxiety for the children themselves, who may feel they must constantly live up to these enhanced capabilities. Furthermore, discussions around genetic modification can spark debates about equity and access, as not everyone has the same opportunities for genetic enhancements, potentially leading to feelings of inferiority among those who cannot afford such modifications. The societal divide between those scientists play a crucial role in ethical decision-making, particularly as their work increasingly intersects with societal issues, environmental concerns, and health challenges. In many fields, such as biotechnology, environmental science, and medicine, scientists are not only tasked with advancing knowledge, but also with considering the ethical implications of their findings. They often find themselves at the forefront of debates, where their expertise is essential in guiding policymaking and informing public opinion. For instance, when it comes to climate change, scientists conduct research that highlights the potential impacts of human activity on the planet, and this research is vital for policymakers who are making decisions about how to address climate issues. Moreover, the ethical considerations surrounding emerging technologies, like artificial intelligence and genetic engineering, require scientists to engage in discussions about the possible consequences of their work. They must evaluate the risks and benefits, considering issues such as privacy, consent, and the potential for misuse. In the case of genetic editing technologies, for example, scientists are not only focused on the scientific breakthroughs, but are also asked to reflect on the moral implications of altering human DNA, which raises questions about the nature of life, identity, and the potential for unintended consequences. In many instances, scientists collaborate with ethicists and legal experts to develop frameworks that guide responsible research and innovation. This interdisciplinary approach is essential, as it enables a comprehensive understanding of the societal context in which scientific research occurs. By participating in ethical review boards, scientists help ensure that research proposals meet ethical standards and protect the rights and welfare of research subjects, whether human or animal. This is particularly important in medical research, where ethical oversight is crucial to maintaining trust in the scientific process. Furthermore, scientists often serve as educators, helping to raise public awareness about the ethical dimensions of scientific advancements. By communicating complex ideas in accessible ways, they can engage the community in discussions about science policy, funding priorities, and research ethics. For instance, during public health crises, such as the COVID-19 pandemic, scientists have had to navigate not only the scientific challenges, but also the ethical dilemmas related to vaccine distribution, public health messaging, and individual rights versus collective safety. The role of scientists in ethical decision-making is multifaceted and dynamic, requiring a balance between scientific integrity and social responsibility. As new technologies and discoveries emerge, the ethical landscape continues to shift, making it imperative for scientists to remain vigilant and proactive in addressing these challenges. Their involvement in ethical discussions not only enhances the credibility of their work, but also fosters a culture of accountability and transparency. Global inequities in access to genetic technologies present a complex issue that highlights significant disparities between different regions and populations. In many high-income countries, advancements in genetic technologies, such as gene editing and personalized medicine, are rapidly integrated into healthcare systems. For example, techniques like CRISPR-Cas9 are being utilized to treat genetic disorders, but access to these innovations is often limited to those who can afford them. On the other hand, in low and middle-income countries, the situation is starkly different. Here, limited resources, lack of infrastructure, and insufficient funding for research and development restrict the ability to adopt and utilize these advanced technologies. Moreover, the ethical considerations surrounding genetic technologies further complicate the landscape. In wealthier nations, there are generally more robust regulatory frameworks, which can support the responsible use of genetic modifications. However, in places where regulations are weak, there may be a lack of oversight, raising concerns about the potential misuse of these technologies. This can lead to situations where only a privileged few benefit from genetic advancements, while many remain in the dark, unable to access life-saving treatments or preventive measures. Additionally, the cultural context plays a significant role in how genetic technologies are perceived and utilized. In some societies, there may be considerable skepticism about genetic modifications, stemming from traditional beliefs or fear of unintended consequences. This can create barriers to acceptance and implementation, meaning that even when genetic technologies are available, they may not be embraced by the local population. Furthermore, global health initiatives often focus on immediate public health needs, such as infectious diseases, rather than investing in genetic technologies that could provide long-term solutions. This creates a gap in research funding and infrastructure development, perpetuating the cycle of inequity. The disparity in education also contributes to global inequities in access to genetic technologies. In regions where education systems are underfunded, there is often a lack of training for professionals in genetics, limiting the workforce's ability to engage with new technologies. Without skilled personnel to operate and implement genetic innovations, the benefits of these advancements may never reach those who need them most. As we move forward, it is crucial to address these inequities, ensuring that genetic technologies are accessible to all, regardless of geographic location or economic status. International collaborations, funding for research in underrepresented regions, and public education campaigns can help bridge the gap, allowing for a more equitable distribution of genetic advancements. By fostering an inclusive approach to genetic technologies, we can work towards a future where the intersection of genetics and personal identity is a fascinating and complex topic, one that delves into how our DNA shapes who we are, both physically and psychologically. Every person carries a unique genetic code, which is inherited from their parents, and this code influences a myriad of traits, from eye color and height to susceptibility to certain diseases. As scientists have made advances in genetic research, they have uncovered links between genetics and various aspects of personality, intelligence, and even behavioral tendencies. For example, studies have suggested that certain genetic variants might be associated with traits such as extroversion or conscientiousness, leading researchers to ponder how much of our identity is scripted in our genes versus shaped by our environment and personal experiences. Moreover, the rise of direct-to-consumer genetic testing has opened up new avenues for individuals to explore their ancestry and health risks. By simply providing a saliva sample, people can gain insights into their genetic heritage, discovering connections to distant relatives or uncovering unexpected ethnic backgrounds. This process can be both enlightening and unsettling, as individuals confront the realities of their genetic makeup, which may not always align with their perceived identity. For some, these revelations can enhance their sense of self, while for others, they may challenge long-held beliefs about their family history or cultural identity. Additionally, there is an ongoing debate about the ethical implications of genetic information. As we gain more understanding of the human genome, questions arise regarding privacy, consent, and the potential for genetic discrimination. For instance, if an employer learns about an employee's genetic predisposition to a certain illness, could this information affect hiring or promotion decisions? Furthermore, the concept of genetic determinism, which suggests that our genes largely determine our fate, is being challenged by the understanding that both genetics and environment interact to shape our identities. This leads to a richer conversation about nature versus nurture, raising important questions about individual agency and the role of personal choices in shaping who we become. In the realm of mental health, genetics plays a key role as well, with studies indicating that certain mental health conditions have a hereditary component. Understanding the genetic factors associated with conditions like depression or anxiety can empower individuals to seek appropriate treatment and support, fostering a deeper understanding of their own experiences. However, it also raises concerns about labeling individuals based on their genetic risks, potentially leading to stigma or self-fulfilling prophecies where people may feel defined by their genetic predispositions. As we continue to explore the relationship between genetics and personal identity, the potential for genetic enhancements in sports is a topic that stirs much debate, as it blends science, ethics, and the essence of athletic competition. Imagine a world where athletes could undergo genetic modifications to enhance their physical abilities, such as strength, speed, endurance, and recovery. This idea isn't purely science fiction. Research in genetics has advanced significantly, opening doors to possibilities that were once thought impossible. For instance, scientists have already identified certain genes associated with muscle growth and recovery, like the ACTN3 gene, which affects how our muscles respond to exercise. Some studies suggest that individuals with a specific variant of this gene may have a natural advantage in power sports, such as sprinting or weightlifting. As these discoveries unfold, the prospect of using gene editing techniques, such as CRISPR, to enhance athletic performance becomes more tangible. CRISPR, which allows for precise editing of DNA, could theoretically be used to enhance traits that contribute to athletic prowess. However, the implications of such enhancements are profound and complex. On one hand, genetic enhancements could lead to unprecedented achievements in sports, pushing the limits of human capability, while on the other hand, they raise significant ethical questions. Would such enhancements create an uneven playing field, where only those with access to advanced genetic technologies could compete at the highest levels? Moreover, the health risks associated with genetic modifications cannot be overlooked. Altering genes carries potential side effects that may not be fully understood. Athletes who undergo such procedures might face long-term health consequences, raising concerns about their well-being. Additionally, governing bodies in sports, like the International Olympic Committee, are grappling with how to regulate or ban genetic enhancements. The current anti-doping regulations focus primarily on substances and methods that provide unfair advantages, but as genetic technologies evolve, the rules may need to adapt accordingly. Another aspect to consider is the psychological impact on athletes. The notion of using genetic enhancements may alter the perception of hard work and natural talent. If athletes can enhance their abilities through science, what does that mean for the value of training, dedication, and perseverance? The story of sport has always been one of human endeavor, pushing boundaries through sheer effort. The introduction of genetic enhancements could shift this narrative, potentially diminishing the achievements of athletes who rely solely on their natural abilities. Additionally, public opinion varies widely on the subject. Some people embrace the idea of genetic enhancements as a means of achieving new records and redefining human potential, while others view it as a form of genetic counseling plays an important role in society, as it helps individuals and families understand genetic conditions, assess their risks, and make informed decisions about their health. Genetic counselors are trained professionals who provide information about how genetic disorders are inherited, how they affect individuals and families, and what testing options are available. For instance, a family with a history of a genetic disorder, such as cystic fibrosis or Huntington's disease, might seek counseling to learn about the likelihood of passing that condition to their children. In such cases, genetic counselors can offer insights into the implications of genetic testing, as well as the potential outcomes of different choices regarding family planning. Furthermore, genetic counseling is not limited to inherited conditions. It also extends to individuals diagnosed with cancer, as certain cancers can have genetic links. For example, women with a family history of breast cancer might benefit from counseling to understand the risks associated with BRCA1 and BRCA2 gene mutations, which are known to increase the risk of developing breast and ovarian cancers. Through this process, genetic counselors can help patients navigate the emotional aspects of receiving such information, guiding them in coping with anxiety or uncertainty about their health. Moreover, as advances in genetic research continue to unfold, the role of genetic counseling is expanding into areas like pharmacogenomics, where understanding a person's genetic makeup can help determine the most effective medications for them, reducing the trial and error approach often associated with prescribing. This personalization of medicine is becoming increasingly significant in improving patient outcomes, as tailored treatments can lead to more effective management of various health conditions. Additionally, genetic counselors are advocates for ethical considerations in genetics, ensuring that patients receive information that is clear and unbiased, especially in discussions about genetic testing and its implications. They work to ensure that patients understand their rights, including the importance of consent and the potential impact of genetic information on insurance and employment. Overall, the impact of genetic counseling is profound, as it not only empowers individuals with knowledge, but also shapes the future of healthcare, promoting a more informed and health-conscious society. Bioethics, a field that explores the ethical implications of biological and medical research, plays a crucial role in genetic research on human subjects, as it addresses the moral questions that arise when scientists manipulate genetic material, whether for therapeutic or research purposes. One of the key principles in bioethics is respect for persons, which emphasizes informed consent, meaning that individuals must be fully aware of what participation in a study entails, including any risks involved. This is especially important in genetic research, where the potential consequences of altering genes can be profound, not just for the individual, but for their descendants as well, raising concerns about long-term effects and the nature of genetic inheritance. Another significant aspect of bioethics in this context is the concept of justice, which relates to fair distribution of the benefits and burdens of research. This becomes particularly pertinent when considering vulnerable populations who may be at risk of exploitation in research settings, especially if they lack the resources or knowledge to fully understand the implications of genetic studies. Researchers must ensure that these groups are not disproportionately targeted for research, and that they receive equitable access to the benefits that arise from new genetic technologies. Moreover, there is the principle of beneficence, which obligates researchers to maximize potential benefits while minimizing harm. In genetic research, this includes not only physical harm, but also psychological and social repercussions that can stem from genetic testing, such as anxiety over potential health issues or discrimination based on genetic information. As scientists explore the complexities of the human genome, they must navigate these ethical waters carefully, ensuring that their work contributes positively to society. The advent of technologies like CRISPR-Cas9, which allows for precise editing of DNA, has sparked significant debate within bioethics, as it opens the door to possibilities such as gene therapy for genetic disorders, but also raises concerns about designer babies, where parents might select traits for their children, blurring the lines of natural selection and raising ethical questions about what it means to be human. The potential for unintended consequences, both in the individual and on a societal level, heightens the need for robust ethical guidelines and regulatory frameworks that can keep pace with rapid advancements in genetic technology. Furthermore, the global nature of genetic research adds another layer of complexity to bioethics, as different countries have varying regulations and cultural attitudes towards genetics. This disparity can lead to ethical dilemmas, particularly when research is conducted in developing countries where oversight may be lax, and participants may not fully comprehend the implications of their involvement. Genetic engineering, a rapidly advancing field, has significant implications for biodiversity, which refers to the variety of life forms in a particular habitat or ecosystem. One of the most notable impacts of genetic engineering is its potential to enhance agricultural productivity. By modifying crops to be more resistant to pests, diseases, and environmental stresses, scientists can create varieties that yield more food, which is crucial as the global population continues to rise. However, the introduction of genetically modified organisms, or GMOs, into the environment raises concerns about their long-term effects on native species and ecosystems. For instance, when genetically modified crops crossbreed with wild relatives, it can lead to the spread of modified genes into wild populations, potentially altering their natural characteristics and disrupting local ecosystems. Moreover, the use of genetically engineered crops often leads to a reduction in genetic diversity among agricultural species, as farmers may favor a few high-yield varieties over traditional seeds. This shift can make crops more vulnerable to pests and diseases, as a lack of genetic diversity means that there are fewer traits available for the plants to adapt to changing conditions. In some cases, this can lead to monocultures, where a single crop species dominates an area, which decreases resilience against environmental changes and increases the risk of crop failure. On the other hand, genetic engineering also has the potential to aid in the conservation of endangered species. For example, scientists are exploring the possibility of using genetic techniques to increase the population size and genetic diversity of threatened animals by creating genetically diverse offspring in captivity, which could then be reintroduced into their natural habitats. This approach, however, must be carefully managed to avoid unintended consequences, such as introducing new diseases or disrupting existing ecosystems. Another significant aspect of genetic engineering's impact on biodiversity is its role in bioremediation, a process where organisms are genetically modified to clean up polluted environments. For instance, certain bacteria have been engineered to break down oil spills or absorb heavy metals from contaminated soils, helping to restore ecosystems that have been damaged by human activity. While this can be beneficial, there is ongoing debate about the long-term effects of introducing these modified organisms into the wild. Additionally, the ethical implications of genetic engineering cannot be overlooked. There is a growing concern about the ownership of genetic resources, as companies patent genetically modified seeds and organisms, which may limit access for small farmers and developing countries. This raises questions about equity and justice in the distribution of technological benefits, particularly in relation to preserving biodiversity and ensuring food security. In conclusion, synthetic biology is an exciting and rapidly evolving field that combines principles from biology, engineering, and computer science to redesign and create new biological parts, devices, and systems. Imagine being able to design organisms with specific traits, or even create entirely new life forms, all through the manipulation of DNA and cellular processes. This field has the potential to revolutionize medicine, agriculture, and environmental science. One of the most promising applications of synthetic biology is in the development of new therapies for diseases. Researchers are exploring ways to engineer bacteria to produce pharmaceuticals or to enhance the immune system's ability to fight off infections. For example, scientists are working on genetically modified T cells that can better target and destroy cancer cells, providing a more effective and personalized treatment for patients. In agriculture, synthetic biology could lead to the creation of crops that are more resilient to climate change, pests, and diseases, reducing the need for chemical fertilizers and pesticides. By engineering plants to fix their own nitrogen or to use water more efficiently, we could help ensure food security while also protecting the environment. Additionally, synthetic organisms could be designed to produce biofuels or biodegradable plastics, offering sustainable alternatives to fossil fuels and reducing plastic pollution. In environmental science, synthetic biology holds the potential for bioremediation, where engineered microbes could be used to clean up oil spills and remove heavy metals from contaminated sites. These microbes could be programmed to thrive in specific environments, breaking down pollutants into harmless byproducts, thus restoring ecosystems that have been damaged by human activity. Moreover, the possibilities extend to bio-based manufacturing, where synthetic biology enables the production of materials and chemicals through biological processes, leading to a more sustainable and circular economy. However, with the immense potential of synthetic biology comes ethical considerations and safety concerns. The ability to create and modify life raises questions about biosecurity, environmental impact, and the moral implications of altering living organisms. Regulatory frameworks are still being developed to ensure that synthetic biology applications are safe for both humans and the planet. As researchers and policymakers work together to navigate these challenges, it is essential for the public to be informed and engaged in discussions about the direction of synthetic biology. Looking ahead, The future applications of synthetic biology are vast and transformative. The integration of artificial intelligence and machine learning into the design process of synthetic organisms could accelerate discoveries and innovations, allowing for the rapid prototyping of new biological systems that can adapt to changing conditions. As we continue to explore the capabilities of synthetic biology, we may find ourselves on the brink. DNA, the molecule that carries genetic information, plays a significant role in shaping not only our physical traits, but also our behaviors, influencing how we interact with the world around us. This relationship between DNA and behavior is complex, as it involves an intricate interplay of genetics, environment, and experiences. Research has shown that certain genes can affect brain chemistry, impacting traits such as temperament, aggression, and even social behaviors. For instance, variations in the MAOA gene, often referred to as the warrior gene, have been linked to aggressive behavior, suggesting that individuals with certain genetic markers may be more prone to aggression under specific circumstances, although environment and upbringing also play crucial roles in shaping these behaviors. Moreover, studies in behavioral genetics have revealed that heritability estimates for traits like intelligence, anxiety, and even certain personality characteristics can be significant, indicating that DNA does contribute to these aspects of behavior. However, it's important to note that heritability does not mean determinism. Rather, it suggests that genetics can predispose individuals to certain behaviors, such as family dynamics, education, and social interactions, can significantly influence how those genetic predispositions are expressed. Interestingly, research also indicates that epigenetics, the study of how environmental factors can change gene expression without altering the DNA sequence, plays a crucial role in this relationship. For example, experiences such as trauma or stress can lead to chemical changes that affect how genes are expressed, potentially influencing behavior over a lifetime. This means that our experiences can literally shape our biological makeup, further complicating the simple idea of a direct link between DNA and behavior. Additionally, studies involving identical twins who share the same genetic makeup have shown fascinating insights into the nature versus nurture debate. While twins often exhibit similar behaviors, differences can arise due to their unique life experiences, demonstrating that while DNA sets the stage for behavior, the environment can dramatically alter the performance of that genetic script. Furthermore, the influence of culture on behavior also cannot be overlooked. Genetic predispositions may interact with cultural norms and values, leading to variations in behavior among different populations. For example, collectivist societies may promote behaviors that prioritize group harmony and cooperation, while individualistic cultures may encourage self-expression and competition, leading to different behavioral outcomes shaped by both genetics and cultural context. In summary, the relationship between DNA and behavior is a dynamic and multifaceted area of study, where genetics provides a foundation, but environmental influences and personal experiences mold the final expression of behavior. Understanding genetic destiny refers to the idea that an individual's genetic makeup, or DNA, significantly influences their traits, behaviors, and even life outcomes. This concept has been a topic of interest for many years, as scientists and researchers explore the extent to which our genes determine who we are. For instance, studies have shown that genetics can play a role in various aspects of our lives, from physical characteristics, such as height and eye color, to more complex traits like intelligence and personality. One fascinating area of research in this field is the role of genes in the development of certain diseases. Conditions like diabetes, heart disease, and various forms of cancer have been linked to genetic predispositions, which means that individuals with specific genetic markers may have a higher risk of developing these ailments compared to others. However, it's essential to note that genetics is not the sole factor at play. Environment, lifestyle choices, and social influences also interact with our genetic makeup. For instance, while someone may have a genetic inclination towards obesity, factors such as diet, exercise, and overall lifestyle can greatly affect whether they actually become obese or not. This interplay between genes and environment is often referred to as the nature versus nurture debate, highlighting the complexity of human development. Moreover, recent advances in the field of epigenetics have further complicated our understanding of genetic destiny. Epigenetics studies how environmental factors can influence gene expression without altering the DNA sequence itself. This means that our experiences, such as stress, diet, and exposure to toxins, can affect how our genes function, potentially altering our health outcomes and even those of future generations. As we delve deeper into the concept of genetic destiny, it's intriguing to consider the implications of genetic testing. With the rise of direct-to-consumer genetic testing kits, individuals can gain insights into their genetic predispositions to certain traits and diseases. While this information can empower people to make informed health choices, it also raises ethical questions about privacy, genetic discrimination, and the psychological impact of knowing one's genetic risks. Additionally, the idea of genetic determinism, the belief that our genes completely dictate our fate, has faced criticism. Many experts argue that this belief oversimplifies the complex interactions between genetics and environment and may lead to fatalism, where individuals feel helpless in the face of their genetic predispositions. In recent years, discussions around genetic destiny have also expanded to include topics like genetic engineering and gene therapy. With the advent of CRISPR technology, scientists can now edit genes with remarkable precision. Eugenics, a term that derives from the Greek word meaning good birth, emerged in the late 19th and early 20th centuries as a movement aimed at improving the genetic quality of the human population. The concept gained traction through the work of influential figures like Francis Galton, who is considered the father of eugenics, and his cousin, Charles Darwin, whose theories of evolution provided a scientific backdrop for these ideas. Initially, eugenics was seen as a way to promote public health, reduce poverty, and enhance societal well-being, with advocates believing that human traits, including intelligence and behavior, could be inherited and improved through selective breeding. In the early days, eugenics had a significant following in many countries, including the United States and Germany. In the US, organizations like the Eugenics Record Office were established to study heredity and promote eugenic policies, leading to the implementation of measures such as forced sterilizations of individuals deemed unfit to reproduce, including those with mental disabilities and various marginalized groups. This movement found support among some prominent scientists, politicians, and social reformers, who believed that controlling reproduction could lead to a better society. As the movement progressed, it became intertwined with social Darwinism, which misapplied Darwin's theories to justify racial superiority and discrimination. This dangerous association culminated in the horrific practices seen during the Nazi regime, where eugenics was used to justify the systematic extermination of millions deemed racially inferior, including Jews, Romani people, and individuals with disabilities. This dark chapter in history caused widespread condemnation of eugenics, revealing the ethical and moral implications of attempting to control human genetics through such extreme measures. After World War II, the horrors associated with eugenics led to a significant retreat from the movement. The Nuremberg trials highlighted the need for ethical considerations in scientific research and human rights, prompting a re-evaluation of eugenics practices worldwide. However, discussions about genetics and heredity continue to evolve with the advent of modern genetics and biotechnology. Today, while the term eugenics is largely viewed negatively, the underlying ideas about genetic selection have resurfaced in different forms, particularly with advances in genetic engineering and reproductive technologies. The historical perspectives on eugenics serve as a cautionary tale, reminding us of the potential consequences when science and ideology intersect without moral consideration. As the field of genetics continues to advance, it is crucial to reflect on the past, ensuring Artificial intelligence, or AI, is playing a transformative role in the field of genetic research, revolutionizing how scientists understand and manipulate genetic information. The sheer volume of genetic data generated by modern sequencing technologies is staggering, with whole genomes producing millions of data points. Analyzing this data by hand would take an unimaginable amount of time, but AI algorithms can process it quickly, identifying patterns and correlations that might be missed by human researchers. For instance, machine learning techniques can predict how specific genetic variations influence diseases, leading to personalized medicine, where treatments can be tailored to an individual's genetic makeup. Moreover, AI is being used to enhance gene editing technologies like CRISPR, which allows for precise modifications of DNA. By employing AI, researchers can better predict the outcomes of these edits, minimizing unintended consequences and maximizing effectiveness. This predictive capability is crucial, especially in fields like agriculture, where genetically modified organisms are developed to be more resistant to pests or to have higher nutritional value. Additionally, AI helps in drug discovery, as it can analyze genetic information to identify potential drug targets. By understanding the genetic basis of diseases, AI can assist in finding new compounds that may work effectively against specific conditions. This process, traditionally time-consuming and expensive, can be accelerated significantly through AI's ability to sift through vast datasets and recognize promising leads. Another fascinating application of AI in genetic research is in the study of evolutionary biology. By analyzing genetic sequences from various species, AI can help reconstruct evolutionary relationships, providing insights into how species have adapted over time. This can span from understanding the genetic basis of traits in specific environments to tracking how certain genes propagate through populations across generations. Furthermore, AI is also enhancing the accuracy of genetic testing. Companies that provide genetic testing services are leveraging AI to interpret results more reliably, ensuring that individuals receive accurate information about their genetic predispositions. This is particularly important in identifying hereditary diseases, where early detection can lead to better management and outcomes. In clinical settings, AI is being integrated into decision support systems, helping healthcare professionals interpret genetic data and make informed choices about patient care. By combining AI with genomic data, doctors can gain a more comprehensive view of a patient's health, leading to better treatment strategies and outcomes. Lastly, AI fosters collaboration across disciplines, encouraging partnerships between geneticists, computer scientists, and clinicians. This interdisciplinary approach is crucial, as it combines expertise from various fields to tackle complex genetic problems, ultimately leading to a better understanding of genetics and improved health solutions. As AI, genetic engineering is a fascinating field that has captured the attention of scientists, ethicists, and the public alike, particularly when it comes to its potential impact on aging, which is a universal experience that affects everyone. Researchers have been exploring how genetic modifications could potentially slow down the aging process, or even reverse some aspects of it, by targeting the genes associated with cellular repair, regeneration, and longevity. One of the most exciting areas of study involves telomeres, which are protective caps at the ends of our chromosomes. As we age, telomeres shorten, leading to cellular aging and eventually cell death. Some scientists believe that, by using genetic engineering techniques, we may be able to extend the length of telomeres, thereby promoting healthier aging and potentially increasing lifespan. Additionally, there are ongoing studies into the role of specific genes, like the sirtuin genes, which are thought to play a role in regulating metabolism and stress responses, suggesting that modifications to these genes could enhance longevity. However, with the promising possibilities of genetic engineering in aging, come significant risks and ethical concerns that must be carefully considered. One major concern is the potential for unintended consequences, as modifying genes can have unpredictable effects on an organism's biology. For example, altering the genetic pathways that control aging could inadvertently lead to an increased risk of cancer, since many of the same pathways are involved in tumor suppression. Furthermore, there are ethical implications surrounding access to these technologies, as they may only be available to those who can afford them, potentially leading to greater inequality in health outcomes. There are also worries about the long-term effects on the gene pool, as making changes to human genetics could have repercussions for future generations. As scientists continue to explore the potential of genetic engineering in the field of aging, they face the challenge of balancing innovation with caution. Regulatory frameworks will need to evolve alongside these technologies to ensure that research is conducted responsibly, with appropriate oversight to protect public health. In the meantime, researchers are also studying natural compounds that may mimic the effects of genetic modifications, such as resveratrol found in red wine, which has been shown to activate certain longevity genes. This approach could provide a safer alternative to direct genetic interventions, allowing individuals to enhance their health and longevity without the associated risks of genetic engineering. Ultimately, while genetic engineering holds great promise for addressing the challenges of aging, it is essential to proceed with care, considering both the scientific possibilities and the ethical, societal implications of such powerful technologies. Throughout history, genetic experiments have provided valuable lessons, shaping our understanding of biology, ethics, and the potential implications of manipulating life itself. One of the most notable lessons comes from the early work of Gregor Mendel, who, through his meticulous studies of pea plants, established the foundational principles of heredity. Mendel's experiments revealed how traits are passed from one generation to the next, laying the groundwork for modern genetics. However, it wasn't until the 20th century, with the discovery of DNA structure by Watson and Crick, that the true complexity of genetic information began to unravel, leading to a deeper understanding of how genes function and interact. Fast forward to the mid-20th century, and we see the rise of genetic engineering, which brought forth both exciting possibilities and ethical dilemmas. The introduction of recombinant DNA technology allowed scientists to splice genes from different organisms, creating genetically modified organisms, or GMOs. While this innovation promised advances in agriculture and medicine, it also raised significant concerns regarding biodiversity, ecological balance, and food safety. These early experiments taught us that, with great power comes great responsibility, highlighting the need for regulatory frameworks to ensure safe and ethical practices in genetic manipulation. One particularly poignant example of the ethical implications of genetic experimentation is the case of the HeLa cells, derived from Henrietta Lacks without her consent in the 1950s. These cells became one of the most important tools in medical research, leading to breakthroughs in cancer treatment, vaccine development, and more. However, the lack of informed consent and the exploitation of Lack cells sparked an ongoing conversation about medical ethics, racial inequities, and patients' rights, emphasizing the importance of transparency and consent in scientific research. Additionally, the infamous Tuskegee syphilis study serves as a stark reminder of the potential for abuse in medical experimentation. Conducted between 1932 and 1972, this study involved the unethical treatment of African American men with syphilis, who were misled and denied treatment in order to study the disease's progression. This horrific chapter in medical history has instigated reforms in ethical standards for clinical trials, reinforcing the necessity of prioritizing the welfare of participants and ensuring justice in research practices. As we delve into more recent advancements, such as CRISPR technology, we are reminded of the lessons learned from the past. CRISPR allows for precise editing of genes, offering incredible potential in treating genetic disorders. But it also poses ethical questions regarding its use. The debates surrounding genetic modification and the question of whether it constitutes playing God is a complex and nuanced topic, deeply rooted in cultural, ethical, and scientific discussions. At the heart of this debate is the advancement of genetic engineering technologies, such as CRISPR, which allows scientists to edit genes with unprecedented precision. This capability has opened up exciting possibilities for treating genetic disorders, enhancing agricultural productivity, and even combating climate change. Yet, it has also sparked significant concern regarding the moral implications of altering life at its most fundamental level. Many people, particularly within certain religious and cultural frameworks, argue that genetic manipulation encroaches upon the natural order, suggesting that humanity should not interfere with the genetic codes that have evolved over millions of years. This perspective raises questions about the sanctity of life and whether humans have the right to make decisions that could drastically alter the course of nature. On the other hand, proponents of genetic research often emphasize the potential benefits, arguing that the ability to edit genes can lead to groundbreaking medical treatments, such as curing inherited diseases like cystic fibrosis or sickle cell anemia. They contend that utilizing these technologies responsibly can improve human health and well-being, and ultimately, that rejecting such advancements may hinder progress. The cultural backdrop plays a significant role in shaping these viewpoints. For instance, in some cultures, there is a strong belief in the value of scientific progress and innovation, viewing genetic modification as a natural extension of humanity's quest for knowledge. Conversely, other cultures may prioritize traditional beliefs and practices, leading to wariness about technologies that seem to challenge established norms. Moreover, the debate often extends to issues of equity and access, as genetic technologies may be expensive and not equally available to all populations, potentially exacerbating existing health disparities. This concern brings in the idea of genetic privilege, where only those with resources can benefit from genetic advancements, sparking discussions about fairness and justice in healthcare. Additionally, the implications of genetic editing extend beyond human health to environmental and agricultural contexts, where genetically modified organisms, GMOs, are used to increase crop yields and resistance to pests. While this can help address food security, it also raises questions about biodiversity, ecological balance, and the long-term effects of introducing engineered species into the natural world. The ethical considerations of genetic playing God are further complicated by the concept of designer babies, where parents might choose specific traits for their children, such as intelligence or physical appearance. This prospect raises significant moral dilemmas about the definition of normalcy. Societal genetic engineering, a field that combines biology and technology, has made significant strides in recent years, impacting global health in various profound ways. By altering the DNA of organisms, scientists can develop new treatments and preventive measures for diseases that have long plagued humanity. For instance, genetically modified organisms, or GMOs, have been used to create crops that are resistant to pests and diseases, which, in turn, improves food security and reduces the reliance on chemical pesticides. This is particularly important in developing countries, where agricultural challenges can lead to malnutrition and food shortages. Moreover, genetic engineering plays a crucial role in the development of vaccines. The use of recombinant DNA technology has allowed researchers to produce vaccines more efficiently and with fewer side effects. A prime example is the rapid development of mRNA vaccines for COVID-19, which were created using genetic engineering techniques. These vaccines not only protect individuals from the virus but also help curb the spread of the disease, showcasing how genetic engineering can respond to global health crises effectively. In addition to vaccines, genetic engineering has opened up new avenues in gene therapy. This technique has shown promise in treating genetic disorders, such as cystic fibrosis and hemophilia, where correcting the faulty genes can lead to significant health improvements. By targeting the root cause of these diseases, gene therapy has the potential to change lives, offering hope to patients who previously had limited treatment options. Another area where genetic engineering is making an impact is in the field of personalized medicine. By analyzing a patient's genetic makeup, doctors can tailor treatments that are specifically designed for their unique genetic profile. This approach can lead to more effective treatments with fewer side effects, as it allows for a more precise match between the medication and the patient's genetic characteristics. As our understanding of genetics continues to grow, the potential for personalized medicine to transform healthcare becomes increasingly apparent. However, the advancement of genetic engineering is not without its controversies and ethical concerns. Issues such as genetic privacy, the potential for designer babies, and the long-term effects of genetic modifications on ecosystems and human health are topics of ongoing debate. It is essential for scientists, policymakers, and the public to engage in discussions about the ethical implications of these technologies, ensuring that they are used responsibly and equitably. Additionally, the global health landscape is influenced by genetic engineering in the fight against infectious diseases. Techniques such as CRISPR gene editing are being explored to create, as we venture further into the cosmos, the future of genetic engineering in space exploration is becoming an intriguing topic, with the potential to transform not only how we travel beyond our planet, but also how we sustain life in the harsh environments of space. Scientists are exploring ways to modify the genetic makeup of plants, animals, and even humans to adapt to the unique challenges presented by space travel, such as radiation exposure, microgravity, and limited resources. For instance, researchers are investigating how genetically modified plants could grow in Martian soil, which is known to be harsh and lacking in essential nutrients. By altering the genetic structure of these plants, scientists hope to enhance their ability to survive and thrive, providing fresh food for astronauts on long missions. Moreover, genetic engineering could play a crucial role in developing bioregenerative life support systems. These systems aim to create self-sustaining ecosystems where genetically engineered organisms can recycle waste, produce oxygen, and even generate food, mimicking Earth's natural processes. For example, scientists are looking at bacteria that can break down waste products into usable nutrients, or algae that can efficiently produce oxygen and absorb carbon dioxide. This could be particularly vital for long-duration missions, such as trips to Mars, where resupplying from Earth would be impractical. In addition to plants and microorganisms, the potential for genetic modifications in humans is a topic of much debate. As space travel becomes more common, the idea of altering human DNA to better withstand the conditions of space, such as increased radiation exposure and muscle atrophy caused by microgravity, is gaining traction. Researchers are studying the genetic factors that contribute to human resilience in extreme environments, hoping to identify ways to enhance our natural defenses. However, this aspect of genetic engineering raises ethical questions about the implications of modifying human beings for space travel, as well as the long-term effects of such changes. Another exciting frontier is the concept of creating entirely new life forms that could be designed specifically for space exploration. Synthetic biology is an emerging field that allows scientists to engineer organisms with tailored functions, potentially leading to the creation of microbes that can produce useful materials, like biodegradable plastics, or generate energy from the limited resources available in space. This could open up new avenues for building infrastructure on other planets, using locally sourced materials rather than relying on shipments from Earth. Furthermore, as we delve into the future of genetic engineering in space, we must also consider the implications for planetary protection. The risk of contaminating other celestial bodies with Earth-based organisms. Education plays a crucial role in enhancing genetic literacy among individuals, allowing them to understand complex topics related to genetics, DNA, and heredity. As advancements in genetic research continue to unfold, it becomes increasingly important for people to grasp these concepts and their implications for health, society, and ethics. Comprehensive education programs that focus on genetics can help demystify terms like gene editing, CRISPR technology, and genetic predisposition, making them accessible to a broader audience. By integrating genetics into school curriculums, students can learn about these topics from an early age, fostering a generation that is more informed about their own biological makeup and the science behind it. Furthermore, educational initiatives, such as community workshops and online courses, can bridge the gap for adults who may not have had the opportunity to study genetics formally. These resources can empower individuals to make informed decisions regarding their health, especially as genetic testing becomes more prevalent and affordable. As people become more aware of genetic concepts, they can better understand how their lifestyle choices, family history, and even environmental factors can influence their genetic expression. Additionally, education encourages critical thinking about ethical dilemmas associated with genetic engineering, such as the potential for designer babies or the implications of gene therapy. In this rapidly evolving field, the role of education is not just about imparting knowledge, but also about fostering discussions that help individuals navigate the moral and social challenges posed by genetic advancements. This understanding is essential in a world where misinformation can easily spread, and where public perception can shape policies and regulations regarding genetic research and healthcare practices. By promoting genetic literacy, education helps to cultivate a society that is not only knowledgeable, but also engaged in meaningful conversations about the future of genetics and its impact on humanity. Such discussions can lead to informed public policies and responsible scientific practices, ensuring that genetic technologies are used ethically and equitably. Genetic engineering, a rapidly advancing field, holds significant promise in the fight against climate change. As scientists explore ways to modify the genetic makeup of plants, animals, and even microorganisms to enhance their resilience and efficiency in the face of changing environmental conditions. One of the most exciting applications of genetic engineering is the development of crops that can withstand extreme weather, such as droughts or floods, which are becoming more common due to climate change. By altering specific genes, researchers can create varieties of staple crops, like rice and corn, that require less water or can survive in poor soil, helping to ensure food security for growing populations, even as weather patterns become increasingly unpredictable. Moreover, genetic engineering can play a critical role in reducing greenhouse gas emissions, particularly in agriculture, which is a significant contributor to global warming. Livestock, such as cows and sheep, produce methane, a potent greenhouse gas, during digestion. Scientists are working on genetically modifying these animals to reduce methane production, which could substantially decrease the overall emissions from agricultural practices. In addition, genetically engineered microorganisms have been developed to improve soil health, enhance nutrient cycling, and even absorb carbon dioxide directly from the atmosphere, effectively acting as a natural carbon sink. Another fascinating aspect of genetic engineering is its potential to promote the growth of biofuels, which could serve as a cleaner alternative to fossil fuels. By engineering algae or other biomass sources to grow faster and produce more oils, researchers are striving to create sustainable energy sources that can help reduce our reliance on oil and decrease carbon emissions from transportation. Furthermore, genetic engineering can help restore ecosystems that have been damaged by climate change, such as coral reefs, by enhancing the resilience of coral species to warmer waters and acidifying oceans, thus promoting biodiversity and ecosystem stability. On the other hand, while the potential benefits of genetic engineering in addressing climate change are promising, there are also ethical and ecological concerns to consider. The introduction of genetically modified organisms into the environment could potentially disrupt local ecosystems, leading to unintended consequences. Therefore, as this field continues to evolve, it is crucial for scientists, policymakers, and the public to engage in discussions about the responsible use of genetic engineering, ensuring that we maximize its benefits while minimizing risks. This balance is essential in harnessing the power of genetic engineering to create solutions that not only combat climate change, but also promote a sustainable future for all living organisms on our planet. The concept of life, particularly when viewed through the lens of genetics, raises profound philosophical questions that challenge our understanding of existence itself. At its core, life can be defined in various ways, But when we delve into genetics, we encounter a more intricate tapestry. For instance, is life merely the presence of DNA, or does it require additional qualities, such as the ability to reproduce, respond to stimuli, or evolve? This question invites us to consider organisms that do not fit neatly into traditional definitions of life, such as viruses, which possess genetic material and can replicate, but do not carry out metabolic processes on their own. Moreover, the debate extends to the origins of life. How did the first living organisms emerge from non-living matter, and what role does genetic material play in this transformation? Scientists theorize about primordial soup and the RNA world hypothesis, suggesting that RNA molecules, which can store genetic information and catalyze chemical reactions, may have been the precursors to life as we know it. This leads us to ponder whether life began as a simple genetic sequence or as a complex interplay of environmental factors and biochemical reactions. As we explore the genetic definition of life, we must also consider the ethical implications of genetic engineering. With advancements in CRISPR technology, we have the power to edit the genetic code of living organisms, raising questions about what it means to be alive. If we can create synthetic life forms, such as bacteria designed to produce biofuels, does this redefine our understanding of life? Are these creations truly alive, or are they merely machines that operate based on programmed genetic instructions? Another philosophical angle involves the continuity of life and the concept of species. Genetics reveals that all living organisms share a common ancestry, yet the definition of a species can become blurred. What happens when two different species interbreed, producing fertile offspring, as seen in the case of wolves and dogs? This challenges traditional notions of species boundaries and prompts us to question the rigidity of these classifications. Furthermore, the implications of genetic predisposition to certain traits or diseases bring forth questions about free will and determinism. If our genetic makeup influences our behavior and health, to what extent are we responsible for our actions? This dilemma touches on the philosophical debate of nature versus nurture, as we consider how much of our identity is shaped by our genetics versus our environment. Lastly, the advancement of genetic technologies, such as gene therapy, pushes us to rethink the meaning of life in terms of health and longevity.